My name is Dante Bolzan. I'm a PhD student at the University of California in San Diego, and I work in the Dr. Farad Eyes Lab technology. And so today I'm going to be presenting this proceedings called Soffritto, a deep learning model for predicting high resolution replication timing. So there are a couple talks on replication timing, everybody on the same page. Replication timing, or RT for short, is the order by which, sorry. Yeah, maybe a little bit of water. So I replicate their DNA, and yeah, thank you. Replicate their DNA, and as you can see in this GIF here, specific sites within each chromosome initiate replication, and this happens in a chromosome specific manner. And replication timing is not only cell specific, it's also cell type specific. And we care about RT because it correlates well with histone marks and transcriptional activity. Its dysregulation is also associated with disease, in particular cancer. And so how is RT measured? The most commonly used assay is called two-stage RepliSeq, and it quantifies RT genome-wide. And the way it works is you pulse cells with this nucleotide analog called BRDU. You then stain cells with propylium iodide, which is this fluorescent dye that binds to DNA. And so the amount of fluorescence tells you the amount of DNA in each cell. And you can then sort cells using flow cytometry into early and late fractions. And you can then isolate that DNA and sequence reads from the nascent DNA that's synthesized. And you can then obtain early and late reads, which you can then map to a reference genome, compute a coverage for a fixed binning resolution, and then obtain this two-stage RT signal, which you see at the bottom right of this figure. And the x-axis is genomic position, and the y-axis is replication time, with positive values corresponding to early replication and negative values corresponding to late replication. However, there are some limitations of two-stage RepliSeq. So one limitation is that, as you could see from the curve, you're only obtaining a single point for each genomic bin. And so it fails to capture replication that could happen over broader time periods, right? If you have some heterogeneity in a cell population. Another limitation is it struggles to capture these replication pause sites, regions of constant replication, which are thought to initiate or terminate replication locally. And so to address these limitations, another assay called high-resolution RepliSeq was developed, and it's a very similar idea, but now you partition S phase into 16 equally spaced fractions. And so now with this assay, you can then obtain what's called a RepliSeq heat map, which is shown on the right here, with the x-axis genomic position and the y-axis corresponding to now 16 discrete values corresponding to each S phase fraction, ordered from early at the top, S1, to the latest S16 at the bottom. And these values are normalized for each genomic bin to sum to 100. And so each entry can be thought of as the percentage of cells that replicates a given genomic bin for a given S phase fraction. And one thing I want to highlight here is that there is a resolution limit here. So we cannot obtain profiles at a high resolution 50 kb due to the fork speed or of DNA polymerase, which is 1.8 kilobases per minute roughly. And so given 30-minute duration of a single S phase fraction, 50 kilobases of DNA is already replicated essentially. And so pros of two-stage RepliSeq is that it's low cost. Data is widely available in multiple cell lines, but the cause that struggles to capture those RT features that I mentioned. High-resolution RepliSeq, on the other hand, captures more RT features, but it's quite costly and technically challenging, and data is only available for a handful of cell lines. And so we want to ask the question, can we develop a computational model that transforms two-stage RepliSeq into high-resolution RepliSeq? And today there are no computational methods that do this or that predict high-resolution RepliSeq. And so one model that we decided to use is the long short-term memory model, which is a variant of recurrent neural networks. It's called LSTM for short. And the reason why, as I mentioned before, there are these specific origin sites at which DNA begins replicating. And as you can see, it propagates bidirectionally. But also notice that there are regions that are spatially far apart can be replicating at the same time. And so a model that is able to capture short-range and long-range interactions would be useful for modeling replication time. And as you can see in the schematic on the right, LSTM cells, which is the unit of LSTMs, decide how much information to keep from the previous state, how much information to add to the current state, and then how much information to pass on to the next state. And through that, you can learn these long-range and short-range interactions. And so with that, we developed Soffritto. And as you can see here, in addition to two-stage repliseq, we utilize 6 histone marks coded as chip-seq data, as well as GC content and gene density, which is the number of genes overlapping each 50 kb bin. And we take these features and we feed it into our LSTM module, which, as you can see here, is bidirectional, which captures the bidirectional nature of replication timing. And our LSTM module is also stacked into L layers. So each component passes on embedding to the next one until eventually the LSTM embedding is passed to a fully connected layer. And we output our repliseq heat map. Here I've normalized it to sum to 1 rather than 100, just for ease of model training. And I employed a KL divergence loss with L2 regularization to prevent overfitting. And so in terms of data we use to evaluate Soffritto, as I mentioned, 16 fraction replication timing data is only available in these five cell lines, so 3 human, 2 mouse. For those who are unfamiliar, H1 and H9 are 2 human embryonic stem cell lines. HCT116 is a colon cancer cell line. Then you have mouse embryonic stem cells and mouse neural progenitor cells. So all the data I use is publicly available. The histone mark chip-seq data is from ENCODE, 2-stage data from 4DN portal, and then the high-resolution repliseq data from the paper that introduced this assay. In terms of our evaluation strategy, we employed an intra-cell line and cross-cell line strategy. So in the intra-cell line strategy, which is illustrated on the left, for each cell line, we leave 1 chromosome out for validation, 1 for testing. And then in the cross-cell line case, which is on the right, we employ a more stringent evaluation, which is we iteratively leave 1 cell line out during model training, and we test on an unseen cell line and unseen chromosome. And we also have to think about what metrics we use for evaluation, right? So now we have this probability, what you can think of as a probability distribution for each genomic bin, representing the 16 fractions. So we use Spearman correlation, right? So that's capturing how well the model is preserving the order of the S-phase fractions for each genomic bin. KL divergence for overall probability distribution difference. And then this metric, which we call argmax RT fraction, which we defined for this paper. And it's a very simple concept. You take your 16 fraction profile and you output the fraction with the highest value as an integer. And you do that for observed and predicted, and you take the absolute difference. And this gives you kind of an intuitive measure of like how many fractions the model is off. And it ignores some information about the distribution, but at least you're getting an idea of how which fraction is most salient, right? And then finally, we look at 2 metrics that can be reformulated as distances between 2 cumulative distribution functions. And the reason why we use these 2 is that the cumulative distribution function of the 16 fraction RT signal is relevant biologically, right? So what that tells you is for each S-phase fraction, what percentage of cells replicated that bin in that fraction and all earlier fractions. And so Kolmogorov-Smirnov metric, which is equivalent to the largest absolute difference between 2 CDFs. And we also use the Wasserstein distance, which can be formulated as the absolute difference in area between 2 CDFs. And so now with all of these metrics and all of this data, we can then run Soffritto in using our intra-cell line evaluation strategy. And so as you can see on the figure on the left, this is the observed and predicted repliseq heat maps for mouse embryonic stem cell line. This is chromosome 9. And as you can see, Soffritto is capturing the general pattern across the chromosome pretty well, but it's also capturing regions that have very specific time frames in which they're replicating, but also bins with a broader time scale as well. And then on the right, so given that there are no computational models that predict high resolution replication time, we decided to use these 5 baseline models, non-recurrent baseline models, as a way to contextualize Soffritto's performance. And so they are a support vector regression model, linear regression, random forest, lasso, and a two-layer neural network. And as you can see from this figure, which is measuring Spearman correlation across the bins for each of the intracell line splits, Soffritto is the best performing model with an average Spearman correlation of roughly 0.95. Now, there are 4 other metrics, and so we decided to use what's called a board count, which comes from voting theory in the social sciences, to give an overall ranking of the different models. And it's a very simple concept. You just rank the models for each metric and cell line, and you assign points to each model. So 0 points to the lowest ranked model, 1 point to the next lowest ranked model, and so on and so forth. And so given that there are 5 metrics, 5 cell lines, and 6 models in total, you can get the largest number of points a model can get is 125 if you sum it up across all of the different splits. And as you can see here, Soffritto is the best performing model with a Borda count of 122. Now, moving beyond just accuracy, we wanted to see if Soffritto is capturing features that you can define using high-resolution RepliSeq data. And so the 2 I'm going to illustrate here are T-rep and T-width. So as you can see on the figure on the top, T-rep is the point along the cumulative replication fraction curve, which 50% of cells have replicated that specific bin. So this is for a specific genomic bin. And then T-width is the amount of time between 25% of cells replicating that bin and 75% of cells replicating that specific bin. And as I'm showing the figure on the bottom here, if we look at observed versus predicted T-rep, we see a very high correlation, greater than 0.9 for almost all of the cell lines. So looking more at T-width, I want to introduce another replication timing feature that you can define using 16 fraction or high resolution data, which is called initiation zones, or IZ for short. And what these are is these are regions of constant replication that are thought to initiate replication because they appear as local peaks in the high resolution RepliSeq heat map. And that's illustrated in the bottom figure on the right here. And so you can then classify these initiation zones as early, mid, or late, depending on where they're located, right, along the S phase fraction y-axis. So this example here, this would be an early IZ. And so the IZ region is composed of consecutive bins. And one finding that was noted in the high resolution RepliSeq paper is that IZs, as you go from early to mid replicating IZs, T-width tends to increase. So what that means is for mid replicating IZs, there is an increase in the cellular heterogeneity of replication timing for that for those particular bins. And then as you go from mid replication to late replication, then you see a gradual decrease in T-width. And so that's illustrated with the 2 figures on the left, if you look at the left column of these 2 figures. And what we found was that Soffritto's predictions also followed this pattern, this gradual increase in T-width as you go from early to mid, and a decrease in T-width from mid to late. And we found this in both H1 cell line and the mouse embryonic cell line. Next, we performed a feature ablation analysis. So here what we're doing is we're taking each feature and iteratively removing it and then rerunning Soffritto using the same train test splits. And so in this bar plot that I'm showing here, the y-axis is measuring the essentially the error in the mean argmax RT fraction when you remove each feature individually. And what we found is that removing 2-stage RT led to the greatest drop in performance. And this was followed by removing the histones collectively. And we found this to be consistent across the different cell lines. And, so this is fine. This is interesting, kind of what you would expect. But then we wanted to look at, what do the predictions of these ablated models actually look like, right? So what happens when we actually visualize them? And so we then looked at, this is a 10 megabase per region on chromosome 9 for mouse neural progenitor cells. And so as you can see in the top heat map, which is corresponding to the observed RepliSeq heat map, you see this slight dip in the replication timing profile, which I'm highlighting in red here, around 15 megabase pairs. And you can see that the full model with all of the features doesn't capture it, or at least doesn't capture it as well. And when you remove the histones collectively, you also don't see it being captured. However, when we remove a two-stage RT feature, then we see that the model does capture that blip, suggesting that even though two-stage RT is the most salient feature, the other features in combination with each other, can capture these local changes in replication timing. And so, it might also suggest that the two-stage RT is maybe leading to an over-smoothing for the model in certain local cases. So then we finally looked at our cross-cell line evaluation strategy. So here I'm iteratively leaving one cell line out, right? And if we look at the heat maps on the left, this is now looking at H9, chromosome 9 and H9, we see that the model's still capturing the overall general replication timing trend. But as you can see for the last 15 megabase pairs, it's kind of broadening the time scale of bins where it shouldn't be broader. You have a sharp replication timing profile, as you can see here. We also see from this figure on the right here, which is again the violin plots for Spearman correlation, there's an overall drop in performance for all of the models. And Soffritto does comparatively worse compared to the other models. And if we look at the Borda count ranking, Soffritto is the second best performing model. And now the neural network actually pulls ahead slightly. And so to conclude, the strong performance of these non-recurrent baseline models suggests that the features for each bin have high predictive power, even in the absence of neighboring bins. And another way that Soffritto could be improved in the future is to include more features, perhaps 3D genome features, right? Because these are also very much correlated with replication timing. And then less features could be defined at higher resolution than 50 kb. So even though we have to make these high resolution replication timing predictions at 50 kb resolution, the features could be defined at much higher resolution. This would give us more data to learn from, and you could then perhaps use a more sophisticated model architecture for that. And so with that, I want to thank my advisor, Dr. Ferhat Ay, as well as the NIH and other members of the iLab as well for their feedback, and as well as the NIH for funding. And Soffritto is available on GitHub. I provide the link right here. And yeah, the paper's on bioinformatics. So thank you all for listening.
Thanks very much. We have time for one or two questions. I can shout one. When you look at, when you compare your predictive power, you see that the worst one seems to be for HCT116, right? Which is a cancer cell line, if I remember correctly.
Yes, you're talking about for the leave one cell line out.
But for both evaluation, I think it's the one that performs the worst in a way.
Yes.
What's your interpretation for that? Can it be due to the fact that it's a cancer cell line, so you have non-deployed amplification and so on, and that is messing up the signal, or have you just thought about that?
Yeah, I think that's a good point, especially given that it's the only cancer cell line, right, in the data sets that are available. I mean, one other limitation, right, of using high res or trying to predict high resolution replication timing is we don't have a wide variety of cell lines, right, to learn from. But yeah, I agree that cancer could disrupt any sort of general, I don't know, grammar patterns, right? So it's possible. Yeah, I'm not entirely sure, but yeah, it's possible.
Yeah, one last question.
Thank you for the talk. I was wondering, in your benchmarking, you compared against I guess a different type of machine learning models, right?
Yeah.
I was wondering if you thought about comparing against a baseline that is not like a learning model, just taking the two-stage replication timing measurements and extended it in some naive way.
Yeah, that's a great question. And this is kind of going back to that talk right yesterday about using good baselines. And so one thing that we did was to take each feature, partition into 16 quantiles, and then just order them, and then just predicting the max RT fraction, right, for each one. And if we do that, then yeah, the performance is nowhere near as good as Soffritto, but it actually is comparable to some of the other machine learning models. And that also shows that two-stage RT is indeed the most predictive on its own. It leads to roughly an error of like 1.5 fractions off. So yeah, good question.
Great. I would like to thank again all the speakers of the session. Thank you.